Hello dear students, Assalamu alaikum. In the last lecture, we have discussed about the breathing mechanism. We have already studied about the process of respiration such as ventilation, diffusion and perfusion, including inhalation and exhalation in last lecture. We have also discussed about intrapleural and intrapulmonary pressure. Now in the current lecture is the continuation of the previous one. Here we will study about gas exchange, role of surfactants, transportation of oxygen, carbon dioxide and carbon monoxide in the blood and in the muscles. Also we will discuss about chloride shift, respiratory volumes and capacities, and we'll try to know the factors that control internal and external respiration and the neural control of respiration. So let's start our today's lecture. Now comes how oxygen is transported in the blood. Oxygen is needed for cells, for body cells and tissues to undergo cellular respiration. Glucose in the presence of oxygen make energy, that is the ATP molecule. We know that ATP is an energy molecule, and water and some carbon dioxide. This carbon dioxide is then exhaled out. Oxygen is one of the substances transported with the assistance or help of red blood cells. The red cells contain a pigment which is called hemoglobin, each molecule of which binds 4 oxygen molecules. That is, when hemoglobin binds to the 4 oxygen molecules, here it carries 4 oxygen molecules. Approximately 98% of total oxygen is transported in this way in the blood and is bound to hemoglobin, while only 2% oxygen is dissolved directly in the plasma without the help of any other molecule. Hemoglobin is a metalloprotein with 4 subunits, as we can see here, the 4 subunits, 1, 2, 3, 4 polypeptide chains. It is composed of an iron containing heme group attached to a globin polypeptide chain. One molecule of oxygen can bind to the iron atom of a heme group, giving each hemoglobin the ability to transport 4 oxygen molecules. We can observe here in blue 4 oxygen molecules that are bound to the 4 polypeptide chains. One molecule of oxygen can bind to the iron atom of a heme group, giving each hemoglobin the maximum capacity to transport 4 oxygen molecules. This ability to bind oxygen to each subunit results in the unique sigmoidal shape of the oxyhemoglobin dissociation curve, as we can see here. Various defects in the synthesis or structure of erythrocytes or hemoglobin or the globin polypeptide chain can impair the oxygen carrying capacity of the blood and lead to hypoxia. That is the low concentration of oxygen in the blood. The oxygen molecules are carried to individual cells in the body tissue where they are released ultimately. The binding of oxygen is a reversible reaction. The 4 discs in this diagram, the blue one, they are the part of the molecule where oxygen molecules are bound, while the 4 folded sausages shapes represent polypeptide chains. At high oxygen concentration, oxyhemoglobin forms, but at low oxygen concentration, oxyhemoglobin dissociates to hemoglobin and oxygen. That is, ultimately it releases oxygen. The balance can be shown by an oxygen dissociation curve for oxyhemoglobin. There is the curve of oxyhemoglobin. We can see here as the partial pressure of oxygen increases, the percent saturation of the oxyhemoglobin increases. The curve shows that at relatively low oxygen concentration, there is uncombined hemoglobin in the blood, that is here, and little or no oxyhemoglobin is found in the body tissue, while at relatively high oxygen concentration, there is little or no uncombined hemoglobin. That is, all hemoglobin is converted to oxyhemoglobin in the blood. It is the form of oxyhemoglobin. For example, in the lungs. It is to note that oxygen and carbon dioxide concentration are expressed as partial pressures. and are measured in kPa, the unit here, also called oxygen or carbon tension. The amount of oxygen held by the hemoglobin, that is its saturation level, is normally expressed as a percentage. Oxygen dissociation curve can be used to illustrate Le Chatelier's principle, which states that system in dynamic equilibrium responds to any stress by restoring the equilibrium. For example, shifts in the position of the curve occur as a result of concentration of carbon dioxide or changes in pH. Then, how carbon dioxide is transported in the blood? It's more important it's also very important because there are more than one types of carbon dioxide transportation. When carbon dioxide diffuses into the blood plasma and then into the red blood cells, that is erythrocytes, in the presence of the catalyst carbonic anhydrase. Here a catalyst is required, carbonic anhydrase, that is used to convert carbon dioxide. Here carbon dioxide reacts with water in the erythrocytes and the dynamic equilibrium is established. Here carbon dioxide actually with the help of water is converted to carbonic acid, H2CO3. We can see here the reaction. Carbon dioxide is converted with the help of water and in the presence of carbonic anhydrase is converted to bicarbonate ion. These bicarbonate ions then binds to the hemoglobin directly because and it is very important fact that hemoglobin's tendency to bind with carbon dioxide is much more as compared to the oxygen. Here carbonic acid H2CO3 dissociates to form hydrogen ion and hydrogen carbonate ions. This is also a reversible reaction. That is, this hydrogen carbonate ion can be easily converted to carbon dioxide or the carbonic acid. And dissociated carbonic acid, hydrogen ions and hydrogen carbonate ions exist in dynamic equilibrium with one another. Inside the erythrocytes, that is inside the red portion, negatively charged at CO3 ions diffuse from the cytoplasm to the plasma. This is balanced by diffusion of chloride shift. HCO3 moves from red blood cells towards plasma. As a result, a chloride ion, in order to balance the changes in the charge and the ion change, in order to balance the ion change, a chloride ion moves in. This is known as chloride shift. It means that this process is balanced by diffusion of chloride ions in the opposite direction to the HCO3, maintaining the balance of negative and positive ions either side. This is called chloride shift. The dissociation of carbonic acid increases the ability of the blood. That is, it decreases its pH. Hydrogen ions then react with oxyhemoglobin to release bound oxygen and reduce the acidity of the blood. This buffering action allows large quantities of carbonic acid to be carried in the blood without major changes in blood pH. Now, the hemoglobin is strongly attracted to carbon dioxide molecule. We have already discussed about it. Carbon dioxide is removed to reduce its concentration in the cell and is transported to the lungs where its concentration is lower. This process is continuous since the oxygen concentration is always higher than the carbon dioxide concentration in the lungs. So the opposite true is respiring cells. So if we talk about the carbon dioxide transport, we can observe that 9% of the carbon dioxide is dissolved freely in the blood plasma without the help of any molecule, while 27% of the carbon dioxide is attached to the hemoglobin as carbaminohemoglobin, while 64% as a combination of hydrogen ions dissolved in the blood or hydrogen ions on hemoglobin, as we have already discussed in the next slide. This schematic diagram shows the transportation of carbon dioxide and the oxygen in the red blood cells, plasma, in arterial, proximal tubular cells and proximal tubules, along with the chloride shift. Now comes carbon monoxide transport in the blood. As we know, carbon monoxide is a colorless, odorless and insipidous gas. It has been the main cause of accidental death from poisoning. It is responsible for the largest amount of pollutant launched in the atmosphere, produced by man and nature. The main source is the fossil fuel burning, that is the coal, oil and natural gas, mostly produced by motorized vehicles. In the presence of less oxygen, when there is not sufficient oxygen is present, instead of producing carbon dioxide, carbon monoxide is produced. The human body produces carbon monoxide at low levels. The function of the human respiratory system is to provide oxygen to the tissues and to eliminate the carbon dioxide produced by them. Most of the oxygen transported by the blood is through its reaction with hemoglobin molecules. The transport of carbon monoxide happens in the same way, but its affinity, that is the affinity of carbon monoxide to bind with hemoglobin, is about 250 times higher than that of oxygen. So in the presence of carbon monoxide in the blood, it reduces the capacity to carry oxygen. Therefore, large carbon monoxide concentrations produce the lack of oxygen in the tissues. The bond between carbon monoxide and hemoglobin is called carboxyhemoglobin, COHB, represented by the amount, that is the percent of hemoglobin reacted with carbon monoxide. It is used to relate the carbon monoxide effect in the human body also. It is measured by blood analysis or exhaled air. In the absence of measurement methods, the carbon monoxide can be estimated through mathematical models also. So let's talk about chloride shift occurring in the transportation of carbon dioxide. What is the significance of chloride shift? Now the question arises, what is it actually? It is the movement of chloride ions from plasma into red blood cells as the blood undergoes the transition from arterial to venous gas. The chloride shift is also known as the Hamburger shift. It is the process that takes place in a cardiovascular system where the exchange of bicarbonate ions and chloride ions occur across the membrane of red blood cells or erythrocytes. When actually does the chloride shift occur? Number one, when the chloride ions moves into the erythrocytes. When the chloride ions moves into the erythrocytes and bicarbonate moves out into the venous blood. And second, when chloride ions moves out and bicarbonate ions moves into the pulmonary capillaries. So if we look upon this diagram, this carbon dioxide coming from tissues towards the peripheral capillaries, entering the red blood cells and is convert and with the help of carbonic anhydrase, it is converted to HCO3 ions. As this HCO3 ions moves towards plasma, in order to balance the charge, that is the negative and positive charge concentration, the chloride ions move into the red blood cells. While on the other hand, when bicarbonate ions moves into the red blood cells, in order to balance the charge shift, this chloride ions is moved outwards, that is out of the red blood cells. Here this HCO3 bicarbonate ion, again with the help of carbonic anhydrase, is converted to CO3, which is then uh, expired out or inhaled or exhaled out of the lungs. So both of these sites use chloride shift in order to exchange carbon dioxide gas. What is the mechanism of chloride shift? The byproduct of normal metabolism is carbon dioxide. This byproduct dissolves in the blood plasma and in red blood cell, where carbonic acid is formed due to the hydration of carbonic anhydrase. We have already seen this process in last slides. By the band 3 exchange protein, bicarbonate is pumped out of the red blood cell, as we can see here, and chloride is pumped into it. This mechanism takes place during the circulating time. The reverse mechanism takes place in the pulmonary capillaries. So the both processes, they are occurring in venous capillaries and in pulmonary capillaries. Bicarbonate is pumped back into the red blood cell and chloride is pumped out of red blood cell. Also, carbonic anhydrase converts bicarbonate into carbon dioxide and water. And this carbon dioxide is moved out through the airways or lungs. So, if we talk about the significance of the chloride shift, we can say that the carbon dioxide carrying capacity of venous blood increases because of the chloride shift. Similarly, the unloading capacity of oxygen increases as there is a modulation in the hemoglobin tetramer, and the change in pH is reduced, that is caused due to metabolic byproducts. This is the summary of carbon dioxide transport. If we start from peripheral cells, we can observe that carbon dioxide generated in muscles, tissues, and as a result of other metabolic processes, it this carbon dioxide enters the blood plasma, where almost 10% of this carbon dioxide is dissolved directly into the plasma without help of any chemical or hemoglobin, while remaining carbon dioxide enters the red blood cells, where it takes two ways. Almost 30% of the carbon dioxide is bound with the hemoglobin, while remaining 60% takes the other way. That is, this carbon dioxide is converted into the bicarbonate ions and ultimately with the help of carbonic anhydrase is converted to carb converted to carbonic acid, where chloride shift helps in this conversion. So this is the overall process occurring for the carbon dioxide and the oxygen transport. That is, oxygen coming from lungs enter the blood and goes to tissues for metabolic activities and other processes, and also provide energy to tissues and muscles to work properly for the routine work, while carbon dioxide producing and generating here enters the blood and takes an inverse pathway to the oxygen. That is, it enters the blood with the help of water. This carbon dioxide is converted to H2CO3, that is the carbonic acid, and in this process occurs in red blood cell, and we have already seen that almost 60% of the carbon dioxide, that is the majority of the carbon dioxide, is transported through this mechanism. Here H2CO3 is again converted to HCO3 bicarbonate ions, where this bicarbonate ions transport carbon dioxide towards lungs, and this carbon dioxide is expired out. Gas pressure and respiration. In the lungs, oxygen diffuses out of the alveoli and into the capillaries surrounding the alveoli. Oxygen, about 98% oxygen binds reversibly to the respiratory pigment known as hemoglobin, which is found in red blood cells. Red blood cells carry oxygen to the tissues, where oxygen dissociates from the hemoglobin and diffuses into the cells of the tissues. More specifically, alveolar partial pressure of oxygen is higher in the alveoli, that is 100 millimeter mercury, than blood, where partial pressure of oxygen is just 40 millimeter mercury. Because this pressure gradient exists, oxygen diffuses down its pressure gradient, moving out of the alveoli and entering the blood of the capillaries, where oxygen binds to hemoglobin. At the same time, alveolar partial pressure of carbon dioxide is just 40 millimeter mercury as compared to the blood, where its partial pressure is 46 millimeter mercury. It means that carbon dioxide diffuses down its pressure gradient, that is from blood towards lungs, moving out of the capillaries and entering the alveoli. Oxygen and carbon dioxide moves independently of each other. They diffuse down their own pressure gradient. As blood leaves the lungs through the pulmonary veins, The venous partial pressure is 100 millimeter mercury, whereas the venous partial pressure of carbon dioxide is 40 millimeter mercury. As blood enters the systematic capillaries, the blood will lose oxygen and gain carbon dioxide because of the pressure difference of the tissues and blood. In systematic capillaries, PO2, that is partial pressure of oxygen, becomes 100 millimeter mercury. But in the tissue cells, partial pressure of oxygen is just 40 millimeter mercury here. This pressure gradient drives the diffusion of oxygen out of the capillaries and into the tissue cells. At the same time, blood partial pressure of carbon dioxide, that is the carbon dioxide concentration in blood, is 40 millimeter mercury, and systematic tissue and partial pressure of carbon dioxide is 46 millimeter mercury. The pressure gradient actually drives their carbon dioxide out of the tissue cells and into the capillaries. The blood returning to the lungs through the pulmonary arteries have a venous PO2 40 millimeter and PCO2 46 millimeter mercury. The blood enters the lung capillaries where the process of exchanging gases between the capillaries and alveoli begins again in the same way. So that this diagram shows the partial pressure of oxygen and carbon dioxide changes as blood moves through the body. Respiratory volumes and capacities. Normal breathing moves about 500 milliliter of air with each breath, which is known as tidal volume, TV. Many factors that affect respiratory capacity, for example, a person's size, its sex. For example, we can say that a person having a large height will carry big amount of air as compared to the short ones. Similarly, if we talk about sex, the male can carry more oxygen as compared to the female. It also depends upon age and physical condition of the person. Now, if we talk about some terms, residual volume of air after exhalation, about 1200 milliliter of air remains in the lungs. What is IRV? Inspiratory reserve volume. It is the amount of air that can be taken in forcibly over the tidal volume. It lies usually between 2100 and 3200 milliliter. What is ERV? It's expiratory reserve volume, that is amount of air that can be forcibly exhaled. IRV is related to inspiration, that is related to inhalation, and ERV is related to exhalation. And ERV volume is approximately remains 1200 milliliter. So inspiratory reserve volume, that is IRV, it's the amount of air that can be taken over the tidal volume. Next comes residual volume. It's the amount of air that can be forcibly, it's the air that remain in lung after expiration. There is always some amount of air that remain in the lungs after expiration, so that to avoid collapsing of the lungs. Its volume is also about 1200 milliliter. Then comes vital capacity, the total amount of exchangeable air. So we can define vital capacity as TV, that is the tidal volume, plus IRV plus ERV. It is also known as dead space volume. That is the air, it is the air that remains in conducting zone and never reaches alveoli, but it remains in the trachea and in bronchi and bronchioles. Its amount is almost 150 milliliter just. This is known as dead space volume. Functional volume, that is the air that actually reaches the respiratory zone, usually about 300 milliliter at the same time. Then comes respiratory capacities, which are measured with a spirometer. Different animals have different lung capacities based on their activities. For example, we can say cheetahs have evolved a much higher lung capacity than humans. It helps provide oxygen to all the muscles in the body and allows them to run very fast. Similarly, elephants, they have very slow speed, so they also have a high lung capacity because of their size. In this case, it is not because they run fast, but because they have a large body and must be able to take up oxygen in accordance with their body size. As we have already seen that body size also depends upon the capacity of the air that is taken. Human lung size is determined by genetics, gender and height. At maximal capacity, an average lung can hold almost 6 liters of air. But lungs do not usually operate at maximal capacity. Air in the lungs is measured in terms of lung volumes and lung capacities. As we can see here in this diagram, in this figure, volume measures the amount of air for one function, such as inhalation or exhalation. Capacity is any two or more volumes. For example, we can say how much can be inhaled from the end of a maximal exhalation. Human lung volumes and capacities are shown here in this diagram. The total lung capacity of the adult male is, we have already discussed, it's 6 liter. And tidal volume, it's the volume of air inhaled in a single normal breath. So, inspiratory capacity is the amount of air taken in during a deep breath, and residual volume is the amount of air left in the lungs after forceful respiration. This diagram shows some terms which we have already discussed in the last slides. That is the total lung capacity, which is the almost 6 liter, vital capacity, which is 4800 ml, and inspiratory reserve volume IRV. Normally for an average human it is measured as 3200 milliliter or 3100 milliliter. Tidal volume is 500 milliliter, and expiratory reserve volume ERV, that depends upon exhalation, it's almost 1200 milliliter, and residual volume is 1200 milliliter. This table shows again some terms, their definitions, their volume in number and their equations. For example, tidal volume, the amount of air inhaled during a normal breath, which is 500 milliliter. Expiratory reserve volume ERV, amount of air that can be exhaled after a normal exhalation. IRV, amount of air that can be further inhaled after a normal inhalation. Similarly, there are some definitions for RV, vital capacity, inspiratory capacity, functional residual capacity, total lung capacity, and forced expiratory volume capacity. The volume in the lung can be divided into 4 units: tidal volume, expiratory reserve volume, ERV, inspiratory reserve volume IRV, and residual volume RV. So these 4 volumes are important for an amount of air that enters the lungs. External respiration. In the start of this presentation, we have already studied that there are two types of respiration: external respiration and internal respiration. So after studying all the mechanism of respiration, now we can define both of these terms. What is external respiration? Where oxygen movement into the blood occurs. That is, in this case, the alveoli always has more oxygen than the blood. Oxygen moves by diffusion towards the area of lower concentration, and pulmonary capillary blood gains oxygen. It means that external respiration involves movement of oxygen into the blood from lungs towards blood, and carbon dioxide movement out of the blood towards the lungs. Blood, in this case, blood returning from tissue has higher concentration of carbon dioxide than air in the alveoli. So the pulmonary capillary blood gives up carbon dioxide, and the third process occurs. It's the blood leaving the lungs is oxygen rich, and carbon dioxide poor. So overall, this is the process of external respiration. While in the process of internal respiration, which is also known as gas transportation in the blood, here oxygen is transported in the blood, that is inside red blood cell this process occurs, which is where oxygen is attached to the hemoglobin in the form of oxyhemoglobin, and a small amount of this oxygen is carried by dissolving directly in the plasma. While if we talk about carbon dioxide transport in the blood, most of the carbon dioxide is transported in the plasma is bicarbonate ions, while a small amount is carried inside red blood cells on hemoglobin, but at different binding sites than those of oxygen. So for internal respiration, we can say that it's exchange of gases between blood and body cells and is an opposite reaction to what occurs in the lungs, that is what occurs in the external respiration. So we can see here that the external respiration that takes place at the lungs and internal respiration that takes place at the blood. There are two phases of respiration, from lungs towards blood and from blood towards tissues. So the first one is external and the second one from blood towards tissue, it's the internal one. External respiration, as shown in this diagram, we can observe here that from lungs and alveolar space where oxygen concentration high and carbon dioxide concentration is low, fresh air is entering the blood, and in from passing from capillaries and heart, it goes to tissues where blood supplies all the oxygen content, and at this stage even we can observe that the blood which enter the tissue capillaries has higher concentration of oxygen as compared to carbon dioxide. Now starts internal respiration, that is transfer and exchange of oxygen from blood towards tissues. While coming back from tissues, carbon dioxide now has higher capacity, has higher concentration as compared to oxygen. After passing from internal respiration, the carbon dioxide goes back to blood. It is dissolved in the blood. Passing from heart, it goes to the lungs and is exhaled out. While for internal respiration, there are two phases as we have already discussed, from alveoli towards red blood cells and from red blood cells towards tissues. Now, if we talk about oxygen, we can observe here that oxygen enters from alveoli towards red blood cells, where by combining with hemoglobin in the form of oxyhemoglobin, it is transported towards tissues, where oxyhemoglobin again dissociates and release oxygen that enters the tissue cells, where carbon dioxide is produced. This carbon dioxide, when enters the plasma, combining with water, converts to carbonic acid. This carbonic acid dissociates and release bicarbonate ions. These bicarbonate ions in the plasma again converts to carbonic acid and produce carbon dioxide, which is then transported to alveoli, from blood towards alveoli, and is exhaled in the outside air. Factors influencing respiratory rate and depth. There are some physical factors, for example, increased body temperature, exercise, talking, coughing. They control the rate of respiratory rate. Similarly, there comes volition, that is conscious control of breathing, and emotional factors like singing, crying, laughing also change the respiratory rate. Then comes chemical factors. For example, there are two important factors in this category: carbon dioxide level and oxygen level. If we talk about oxygen level, then changes in oxygen concentration in the blood are detected by chemoreceptors in the aorta and carotid artery, and information is sent to the medulla oblongata. Medulla oblongata in our brain, actually in our brain there are 2 points, medulla and pons, that control the respiratory factors. And if we talk about carbon dioxide level, Level of carbon dioxide in the blood is the main regulatory chemical for respiration. Increased carbon dioxide increases respiration. Obviously, when blood will detect the increase in the concentration of carbon dioxide, it will automatically release the process of respiration in order to avoid the carbon dioxide, extra carbon dioxide in the body. So, similarly, changes in carbon dioxide act directly on the medulla oblongata. Next comes the neural regulation of respiration. The activity of respiratory muscles is transmitted to the brain by the phrenic and intercostal nerves. Neural centers that control rate and depth, they are located in the brain, that is in the medulla. The pons also appears to smooth out respiratory rate. Respiration can be regulated by two mechanisms, namely nervous mechanism and chemical mechanism. Nervous regulation of respiration includes respiratory centers which are present in the medulla oblongata. It detects the level of oxygen and carbon dioxide, and most important, the level of carbon dioxide affects its rate in the blood and sends the required signals by the respiratory muscles. The respiratory centers, they are further divided into two groups, namely medullary centers and pontine centers. The medullary centers have inspiratory centers and expiratory centers. That is, both centers for inspiration and expiration. While the inspiratory center is concerned with the inspiration when the quiet breathing is going on, and the expiratory center is concerned only during forced breathing. While on the other hand, the pontine center have pneumotaxic center and apneustic center. These are located in the pons. The pneumotaxic center controls the activity of respiratory centers, and apneustic centers, they act on the inspiratory centers and increases the depth of inspiration. Now there is a very important and interesting question that arises. The blood level of carbon dioxide gas, the blood level of which gas, oxygen or carbon dioxide, they are important in order to control the nervous mechanism. So the answer is that the blood level of carbon dioxide gas is most important in controlling the respiratory rate. As the chief center respiratory rhythm center located in medulla oblongata is more sensitive for hydrogen ion concentration formed from due to increase in carbon dioxide concentration in arterial blood. So we can say here that there are two pathways of motor neuron stimulation of the respiratory muscles. The first is the control of voluntary breathing by the cerebral cortex, and the second is involuntary breathing controlled by the medulla oblongata. There are chemoreceptors in the aorta, the carotid body of carotid arteries, and in the medulla oblongata of the brain stem that are sensitive to pH. As carbon dioxide levels increase, there is a buildup of carbonic acid which releases hydrogen ions and lowers pH. Thus, the chemoreceptors do not respond to changes in oxygen level, which actually change much more slowly, but to pH, which is dependent upon plasma carbon dioxide level. In other words, carbon dioxide is the driving force for breathing. The receptors in the aorta and the carotid sinus initiate a reflex that immediately stimulates breathing rate, and receptors in the medulla stimulate a sustained increase in breathing until blood pH returns to its normal level. This response can be experienced by running a 100-meter race. During this exertion or any other exercise activity, muscle cells must metabolize ATP at a much faster rate than usual, and thus it will produce much higher quantities of carbon dioxide. So the blood pH drops as carbon dioxide level increase, and we will involuntarily increase breathing rate. Very soon after beginning the sprint, we will continue to breathe heavily after the race, thus expelling more carbon dioxide until pH has returned to its normal level. Metabolic acidosis, therefore, is actually corrected by respiratory compensation, or it's known as hyperventilation. So this is the mechanism by which regulation of blood pH is maintained through respiratory system. This diagram shows the neural regulation of respiration. In brain, we can see here the pons and the medulla oblongata, which are breathing control centers. Both these centers, they send nerve impulses that trigger contraction of muscles. They tend to expand intercostal muscles and diaphragm in order to take more oxygen inside or more air inside the lungs. As soon as more oxygen is absorbed in the blood, heart senses the oxygen concentration in arteries through aortic arch, and it sends message to the breathing control centers in order to control the concentration and the partial pressure of the oxygen concentration. So ultimately, carbon dioxide, when carbon dioxide is increased, so ultimately nerve impulses from oxygen sensors, they indicate the oxygen and decrease its concentration by stopping the inspiration process. While on the other hand, carbon dioxide concentration, when is increased in the blood, it acts directly on the pons, as we have already seen in the, we have already discussed in our talk that carbon dioxide is a driving force for breathing. It directly acts on the pons and medulla oblongata and control the respiration process. Nervous control of breathing by involuntarily way. Involuntary respiration is under subconscious control. The diaphragm and intercostal muscles, they are primary respiratory muscles. They are stimulated by groups of neurons located in the pons and medulla, as we have already discussed last slides. So these neurons from the respiratory control center, they send impulses to the primary respiratory muscle via phrenic and intercostal nerves. We stimulate their contraction. There are three main groups of neurons which are involved in respiration. The ventral respiratory group that controls expiration. These are the ventral respiratory groups that controls expiration, and ultimately it controls the internal intercostal muscles. Then comes the dorsal respiratory group that controls inspiration. So ultimately it affects and controls the diaphragm and external intercostal muscles. And the pontine respiratory group that controls the rate and pattern of breathing. So here are pontine respiratory group. So there are two centers, pneumotaxic center and apneustic center, that control the rate and pattern of breathing. Once the neurons stop firing, the inspiratory muscles relax and expiration occurs. If we talk about breathing rate, the rate at which we inhale and exhale is controlled by the respiratory center. Inspiration occurs due to increased firing of inspiratory nerves and also the increased recruitment of motor units within the intercostal and diaphragm. Our lungs are prevented from excess inspiration due to stretch receptors within the bronchi and bronchioles, which send impulses to the pons when stimulated. While for the process of exhalation, it occurs due to a sudden stop in impulses along the inspiratory nerves. Breathing rate is all controlled by chemoreceptors here within the main arteries, which monitor the level of oxygen and carbon dioxide within the blood. If oxygen saturation falls, ventilation accelerates to increase the volume of oxygen inspired. If we talk about the homeostasis, homeostasis is maintained by the respiratory system in two ways: gas exchange and regulation of blood pH. Gas exchange is performed by the lungs by eliminating carbon dioxide, a waste product given off by cellular respiration. As carbon dioxide exits the body, oxygen needed for cellular respiration enters the body through the lungs. ATP produced by cellular respiration provides the energy for the body to perform many functions, including nerve conduction and muscle contraction. Lack of oxygen affects brain function, sense of judgment, and a host of other problems. So, for breathing and homeostasis, homeostatic change, oxygen in blood increases and carbon dioxide in blood decreases. The initial condition is 12 to 20 breath per minute, but it is triggered and ultimately a heavy and exercised workload brings more oxygen inside. So oxygen in blood decreases ultimately because more oxygen is used and carbon dioxide in blood increased, which is sensed by chemoreceptors in the aorta and other blood vessels respond to increase in carbon dioxide. So ultimately they coordinate their brain control centers or respiratory centers, pons and medulla oblongata, which effect and what it does, the inspiratory nerves fire more often to increase the rate of breathing, causing an increase in the breathing. So our lecture related to respiratory system, gas exchange, functions, how it controls the carbon dioxide and oxygen level ends here. Thank you.